Gebhard from Agilent Technologies. I think he's a product specialist for the Agilent Technologies in Germany. And is going to, the title of this talk is Cancer Stem Cell, Determining Cell Fate of OXPHOS and Glycolysis Measurements.
Yeah, thank you so much for the kind introduction, Christina, and also thank you so much for the invitation to give a presentation here. So when I thought what I could present, I thought I'd make the title first. So the title is CSCs, Determining Cell Fate by OXPHOS and Glycolysis Measurements. And if perhaps already summarizing up a bit the first day of this very nice conference, luckily, I guess this question was already answered by all the other presentators. So if you think about cancer and cancer physiology, also cancer stem cells, they're influenced by a lot of factors like oxygen, nutrient availability, different cells in their surrounding, also immune cells. And somewhere in the middle, you will probably then find the cancer stem cells, which are also influenced by all these factors and adapt their metabolism. And what I think what comes more and more is that you could see a bit of parallels between cancer cell metabolism and also immune metabolism. Because also in immune metabolism, we have different cells, different subsets with different functions, which are also influenced by the microenvironment, of course. And it seems to be the same with the cancer cells, which are not that deregulated as you would have thought first, and just the strong Warburg effect and all these things. So you could really say also in the context of cancer that metabolism not only shows what a cell is, but also what it does and what pathways it's going to use and what it will do. So this can, of course, be influenced by a lot of things like genetic background, nutrient availability, but it seems to be some very specific pathways going on there. So with the CRs, of course, then, as we have also seen in some very nice presentations, of example from Puno Science or Giulio Rossini, we could really see that also with the seahorse, you can measure that quite nicely. So you can use this technology with these sensor cartridges and these measurement cycles to gain information about the metabolic phenotype, about mitochondrial function, glycolytic function, also to see on which substrate oxidation pathways cells might be dependent on, such as glucose oxidation, fatty acid oxidation, glutamine oxidation. So there's a lot you can do with the CRS technology. And when I prepared this presentation, I was thinking, okay, what can I do? I was also going through literature, what was published with the CRS and cancer stem cells. And I saw, okay, with all the important speakers here, they will be able to present the science about the cancer stem cells much nicer than me. So what I thought what I will do is I will show some examples for an assay we have and a way to analyze data, which is not used that much until now, although we have this assay now for about 2 years. So to give you some examples there, and then also to introduce and showcase a new instrument which we introduced in summer to you. And for that reason, I would like now to explain the ATP rate assay. So the ATP rate assay is a kit for the CRS instruments, but it's also a very nice way to look into metabolic data, which you can gain from a CRS experiment. And if you look just into the middle figure there, you can basically also see what an ATP rate assay consists of. We have an injection of oligomycin to see the inhibitory effect on oxygen consumption rate on the respiration in blue. And then we can calculate from the effect of oligomycin the coupled respiration, and from that the micro-ATP production rate. And in the second step, we inject rotin antimycin A, that are complex 1 and complex 3 inhibitors of the mitochondria. By that, we inhibit the complete mitochondrial respiration. the rotenone antimycin A effect then in total from the beginning will give us information about the complete respiration. And this is important then to be able to derive from the ECA, from the extracellular acidification rate, more quantitatively what is really glycolysis going on in these cells. So what we do for the glyco-ATP production rate is then we first translate the ECA, which is the pH change we measure in the system, into milli and picomole per minute secreted protons, into a PR, a proton efflux rate. And then we use the rotenone antimycin A effect and the mitochondrial respiration determination from that to be able to subtract from what we see in acidification, what is the mitoacidification. And this is all the CO2 produced in the mitochondria, secreted into the medium, and also acidify the medium. And if we can subtract that, all which is left in the acidification should then really be the glycolysis of the cells. And then if you do such an assay, the software can nicely calculate for you glyco-ATP production rate, mito-ATP production rate. If you add both together, you have the total ATP production rate. It can give you nice bar graphs as on the upper right, or also you could gain an XF ATP rate index from that, which is just mito-ATP divided by glyco-ATP production. And in the example, you would see here different genetic backgrounds of breast cancer cell lines. We have in blue the estrogen receptor positive, in gray the estrogen receptor negative, and in green some controls. And you could see that basically all the estrogen receptor positive cancer cells are more shifted towards the oxidative side. So they have a specific genetic background, which leads to specific metabolism. In the first real example now with the ATP rate assay, I would like to go again more into the immunology field and really try to explain a bit why I think there are some certain parallels between immune metabolism and cancer cell metabolism. So if you have, for example, a T cell, which is naive, it will be quite quiescent, which you can see that this bar, which you can see next to the cell, is quite small. And it will be quite oxidative, which is shown here or indicated by the large blue proportion. If you activate such a T cell by giving it an antigen-specific activation marker or anti-CD3, anti-CD28 antibodies and make it an effector T cell, it will increase metabolism a lot, which is then shown here by this increased bar for total ATP production. But also the glycolysis ATP production will increase over proportional. So when you drive a naive cell into an effector T cell, it becomes glycolytic, it will proliferate a lot and show a very distinct metabolic phenotype. And then at a later time, usually there can two things happen with these T cells. They can become memory T cells, which means they lower the metabolism again. And also the OXPHOS ATP production plays a larger role then. Or they can, with time, run into exhaustion when they are chronically busy with fighting the pathogen or the cancer cells. And this exhausted cell will be also characterized by a lower metabolism, but this time the glycolytic ATP production will have the reign. And what one big topic, for example, in immunotherapy is at the moment, how could you balance these two metabolic pathways to have fit T cells to fight cancer cells, for example, for a longer time? And here, of course, the question is, we have seen now nice examples from Bruno Science or Julia, really showing how the metabolism changes, how by influencing metabolism, you can also change the direction the cancer cells develop. How would such a map look in the context of normal cancer cells and cancer stem cells? And as in the example in the very first presentation this afternoon, with the generation of cancer stem cells very efficiently with using galactose to make cells oxidative, You can also use something similar to determine the functionality of T cells. So what was done here is T cells were activated with anti-CD3, anti-CD28 antibodies into effector T cells. And when you activate these T cells and you measure that in real time with the seahorse, a significant key event of effector T cell activation is a glycolytic switch where the cells increase their glycolysis. And this is what you exactly can see here on the upper left figure. So we see here the PR as a measure for glycolysis. And when you have the cells as in the red group on glucose, you see there's a nice sudden increase in glycolysis as soon as you activate T cells. If you have them on galactose to drive them in a more oxidative pathway, you can see you don't see that reaction. On the other hand, when you look on the OCAR, you can see that the cells on galactose after activation increase the oxidative metabolism more. And if you then would use an ATP rate assay to calculate what happens with these cells metabolically once they were activated, you can see on the upper right that with glucose or galactose in the medium in the naive T cells, there's not too much of a difference. When you activate the T cells with glucose in the medium, where they can become glycolytic, and you activate them, they will about double their ATP production, but the glyco-ATP production will it will over proportionally increase so that the cells can do the glycolytic shift. When you have them on galactose, they cannot become glycolytic, but they will also about double their ATP production. But now it's just mainly really micro ATP production and OXPHOS driving that. If you look on the lower left, it doesn't change too much in the ATP level. So when you lyse the cells and have a look how much ATP is in them, there's not too much of a change, meaning that they are all nicely in their energetic equilibrium. They produce as much energy as they need. When we would look now into markers of effector T cell activation, such as interferon gamma secretion or IL-2 secretion 2 days after activation, you can basically see that only the cells which had glucose are able to secrete these markers. The cells on galactose don't do that, so they don't become effector T cells. But as we can see from the ATP rate assay, there is something going on, and they were also increasing their metabolism. So what do they do? And I don't have a slide for this, but from other experiments, other publications, we can see that if you activate naive T cells into effector T cells and you prevent them to go into the glycolytic metabolism, they basically directly can quite efficiently form memory T cells, which are again a more long-lasting, so to say, again, more stem-like cell type. And I think this is quite a nice parallel from what we have seen from Bruno Stein's presentation, that you really, with shifting and determining metabolism, you can also determine the cellular outcome. In the other example, which I would like to show with the ATP rate assay, we just had a look how can different genetic backgrounds and different substrates you supply influence cell metabolism. And we were looking here at non-small cell lung cancer cells. And we have 2 different EGFR and 2 different KRAS mutated cell lines on the upper left. And when you look and compare them, you can see in the figure B that the EGFR mutated cell lines are more oxidative. So they gain about 70% of their energy from OXPHOS, whereas the KRAS mutated cells are a bit more glycolytic. So they gain more than 40% from the energy from glycolysis and only about 60% from mitochondrial respiration. And then you could give, as on the right figure, different substrates to see how that changed the energy metabolism. So what we did there is we did give them either 10 millimolar lactate or not, a very low glucose concentration, 0.5 millimolar, or a more physiological glucose concentration, 5 millimolar. When you look at the KRAS mutated A549 cells in figure B, you see there's not really too much of a difference. When you look on these EGFR mutated cells, and especially on the two right bar graphs, you can see we have in this condition both groups in 5 millimolar glucose, but the very right bar, the cells there did get additionally 10 millimolar lactate. And what seems to happen there is that when there is lactate available in a higher concentration, these cells seem to prefer to take up the lactate and to oxidize it. So there's again a metabolic shift towards a more oxidative phenotype with lactate present with this very genetic background of these PC9 cells. Also showing how metabolism can be influenced by different nutrient availabilities. In the last part of my presentation, I want to introduce our new seahorse to you. This is the seahorse XFHS mini analyzer. That's the successor of the XFP. It's an A12 system for which we will introduce very specific plates and also a new analysis software. And what is the special thing with this one compared to all the other seahorses we have, also to the XFEs, is that we did improve sensitivity with hardware and software improvements, and that it will have a much more robust measurement of especially low oxygen consumption rates. So we use really new algorithms there, which then reduce the variation at low OCARs below 40 picomole per minute. And this is what you can basically see on this slide. So when you look at the standard deviation between the XFP and the HS Mini with the same plates, the instrument is much more precise. What we will also introduce then, beginning of next year probably, a bit delayed due to all the COVID troubles, is a new consumable. This is the HS Mini plate. And this mini plate was designed in a very special way that we have a special plate with a ring form, which you can see on the right side, which enables you to really seed the cells more concentrated directly under the sensor, so that in the end you would need less cells for measurement. And as you can see here in a standard XFP plate, you would seed, for example, for C2, C12 cells, 12,000 cells. With the HS Mini plate, you would probably only need about 3,000 cells to generate the same result. So with this new instrument and this new plates, you will be able to reduce cell numbers you would need for an analysis by the factor of 3 or 4. Last but not least is our new software. That's the seahorse analytics software. It's a free software which you can use with all instruments, which is so mandatory for the new HS Mini. You can simply generate an account online for free, upload your data, and there we have really nice and user-friendly new tools to analyze data. So instead of the wave and all these report generators, we have everything together. It's customizable, and you can just generate the data analysis pathways you would like to have saved them, and then always look with your customized analysis into your data. And if you're interested for that, if you're using a seahorse, we can for sure also set up a more specific seahorse analytics workshop for you to explain a bit better how this software works. And with that, I would like to thank you all for attending this presentation and this nice afternoon session. And if there are any questions, I'm open for questions.
Okay, thanks a lot, Daniel, for very nice overview of the features of this new technology. There is a question here asking, thanks for the presentation. Can this experiment be done on XFE24?
You can also use the ATP rate analysis with the XFE24, yes. And also the software.
Okay, I have a question also. I saw your, I mean, you show the application to measure the differentiation of T cells, for example, activation and differentiation. It would be possible to do the similar experiment, but through the co-culture of T cells with target cells, for example, and discriminate the two metabolic.
Yeah. That's not that easy, unfortunately, because we always measure what is in the well together. So you could think if you use only a few T cells and you're interested in the targeted cells, then you could do that and assume that, okay, as you use that few T cells, you won't see the effect of the respiration and acidification from the T cells, and you just see the effect on the target cells. Of course, when it's a mixed signal, you cannot discriminate that in the end. So some people try to circumvent that by having in different groups, just one cell type, then only the other cell type, then a mixture, and try to conclude. But of course, that's then always a thing of interpretation in the end, because you just measure the mixed effect.
Okay, I see. Thank you. There is another question, yes, still from Abiola Bularam Ayanlaya. Also, can the experiment be done if I have oligo and rot, or I have to buy the kit?
So we are, of course, happy when you buy the kit, and we always look for the best quality of the compounds. But in the end, it's an open system. So we won't see if you don't use the kit, and you can do all the analysis and software tools anyways. So in principle, yes, you can also use Sigma compounds also.
He's asking if it's Agilent, I guess the kit.
Yes, so we have the kits from Agilent. You will find them on the homepage where you find the CSCs information. Yeah.
Okay, I don't see other questions. I think if the attendee has still some technical question, scientific question you want to ask, you can still use the chat to submit your question for a few minutes. Since we don't have other questions, I think we can wrap up this session. I would like